In the last part of this lecture series, we're going to talk about the normal processes of neural development and look at critical periods of neural development and how that's related to specific developmental disorders. So early in the development of the embryo, your embryo only has three layers that follow both the cephalocaudal and proximodistal principles, which means they start by developing along the head region, the rostral region, and then develop towards the caudal or tail part. And then things towards the middle, the proximal sections, the medial sections, are developed first, so towards the middle of the body, and then distal limbs are developed second. Your three layers are the ectoderm, mesoderm, and the endoderm. Your ectoderm is your nervous system and skin, which you can see in blue. Your mesoderm is your muscle, bones, and blood vessels, which is in the middle. And then your endoderm will become your internal organs. So in neural development, though, that skin surface will fold in, and so you've got a neural plate which forms from the ectoderm. The neural plate border in green is where it folds inward, and that neural fold will then reseal itself. And then part of that ectoderm will form into a neural groove and then eventually become a neural tube. So your neural tube originates in the central nervous system and it begins forming around 3 weeks after fertilization. And then your neural crest will be what forms part of your spinal nerves, your peripheral nervous system. But sometimes in development, those processes don't develop normally. So when the neural tube is starting to seal, it may not seal completely. In mild cases, you'll develop spina bifida, where the neural tube hasn't completely closed. So that would be what you see in number 1 here. In number 2, you get anencephaly, which is when the brain portion of the central nervous system doesn't form completely. In anencephaly, these embryos usually do not survive to a full-term pregnancy. Again, we're not completely sure about what causes spina bifida and anencephaly, but we do think that it may be related to low levels of folic acid in your diet. Folic acid takes about 8 to 12 weeks to be processed, so you would have to be eating folic acid, which you often find in green leafy vegetables, prior to even getting pregnant. The other thing that's happening early in neural development is what's called neurogenesis. Neurogenesis is the formation of neurons and glia along the ventricles. So you may remember from the brain model activity that when the fetus is starting to form a neural tube into its brain and central nervous system, right, the hindbrain, the midbrain, and the forebrain, during this time you're getting the formation of new neurons and glia along those ventricles, and those are then migrating. So there are radioglial cells here on the edges that are radiating out from the ventricles, and these migrating cells travel along these radioglial cells out to where they need to be. They're guided, and about 2/3 of them move along the radioglial cells, and about the remaining third move in a horizontal fashion. The other thing that you start to get is differentiation of the neural tube along both the dorsal axis, which is your sensory nervous system, and the ventral axis. Remember, ventral is towards your stomach, so all your motor nerves come out of the spinal cord towards your stomach, and the sensory nerves all come out near the dorsal roots. This differentiation also happens from the spinal cord all the way into the mesencephalon. Remember the mesencephalon and metencephalon, you've got cranial nerves that are coming out of these regions. The differentiation also occurs along a rostral caudal axis, and it's influenced by these chemical factors that we've called differentiation-inducing factors. So some of that happens because of internal factors, intrinsic factors, but some of it is also because of environmental signals like folic acid and other chemicals in your diet that help to produce the differentiation of these different nervous system areas. You also have synapse formation. So initially in early fetal development, you don't have a lot of synapses forming between axons and dendrites growing towards one another. But especially after you're born, you get lots of synaptic growth, and that's because these chemical markers will attract axons and dendrites to grow together. And growth cones on the ends of these dendrites stick to nearby cells, and then they pull them along. So these growth cones are essentially inching their way up through the brain until they get to the appropriate synaptic connection for the chemical that they're looking for. And synapse formation also includes the process by tells the neuron what type of neurotransmitter to release. So in addition to growing towards a particular chemical site, along the way, it's also differentiating to release particular kinds of neurotransmitters. So not every neuron releases every kind of neurotransmitter. And chemical signals from the nearby cellular environment are what tell these neurons what kind of neurotransmitter to release. So if you're in an area full of serotonin neurons that release serotonin, those cells are going to tell you, oh, you should also be releasing serotonin. So it's almost like conforming to what every other neuron is doing around you. But we have seen that some of these differences in synaptogenesis may be related to differences in some disorders. So individuals with antisocial personality disorder and schizophrenia actually have poor synaptic formation. So in schizophrenia, there are too many synapses, and in antisocial personality disorder, there are too few synapses in the frontal lobe, suggesting that maybe something is happening early in development that isn't, where they're not getting the right signal. In addition to synaptogenesis and neurogenesis, one of the things that's critical is getting rid of cells that you don't need to use. This process is called cell death or apoptosis. This is when cells go through a process of killing themselves off. And the way this happens is, so one of the other things that's critical, besides forming new neurons and new synapses, you also need to get rid of cells you're not using. We call this process cell death or apoptosis. It's through a process by which a chemical signal causes a cell to suddenly release all of its calcium. This sudden release of calcium causes a lot of the internal workings of the cell to stop functioning, and it causes the cells to not work. Neurotrophins, like nerve growth factor, actually are what tend to prevent cell death. So if cells aren't receiving those neurotrophins, then they're not going to keep functioning. We have seen that children with autism have really high levels of neurotrophins, which might then stop cell death. And one of our theories about autism is that individuals with autism have too many cells, so they're getting overstimulated because they've got too many signals coming in at once. In addition to removing cells you're not using, you also need to get rid of synapses you're not using. We call this process synaptic pruning, where you keep the functional synapses, but any synapses that aren't getting stimulated don't get kept, and they will regress or disappear. And then lastly, we need myelination. Remember, myelin helps to speed up electrical signaling and make it more consistent. Myelin forming along axons starts from the spinal cord first, and then it works its way up, and your sensory neurons are also myelinated first. Research actually early on showed that you could selectively breed for maze running ability in rats. So this is a little pixelated, but it shows actually early studies about in a regular population of rats. Some rats made more maze errors and took them longer to get to the maze. Some made fewer maze errors, and then most of them sat somewhere here in the middle. But if you started selecting two rats, male and female, that made the fewest number of errors, and you bred them together, by the fourth generation, you would have, this is their offspring. So by 4 generations later, we called these maze bright rats. If you take the rats that make the greatest number of errors here and you bred them together, you would get these maze dull rats. By 8 generations, you can see that these two distributions, these are the percentage of the population. You can see these maze bright animals are making very few errors. These maze dull animals tend to make many more errors. Interestingly enough, though, if you were to take those maze dull animals and you raise them, okay, so remember, these are rats who are making lots of mistakes in mazes, and we've selected for this. We've genetically selected for this ability. But now, we take those maze dull rats and we raise them either in these enriched play school toys on steroids environment, this enriched environment, or we raise them in a traditional rat cage which has a running wheel, shavings, food, and water, but nothing else. Then let's see what happens to them. And in fact, in an enriched environment, these rats, these maze dull rats, actually make fewer errors than rats raised in the impoverished environment, right? So this is the number of errors. So these are rats making more mistakes, and these were raised in their typical rat environment. These are enriched rats. They're making fewer mistakes. So even with these maze dull animals, it seems to be that you can turn on these genes that allow them to suddenly be better at exploring their environment, better at running mazes. We know a lot about why that happens. If you actually look at individual neurons within the brains of these rats, you can see, look at the number of branches on these enriched rats' brains. So this is a pyramidal cell that's found in the cerebral cortex of rats. These are isolated animals. You can see that there's not as many connections here in the isolated rats, but there's more in the enriched rats. And if you went actually down to the level of the dendrite, you can see that enriched rats actually have more spines present because they have greater neural connectivity. So the enriched environment is helping them, even in these maze-dull animals. And in fact, when we started to do some genetic experiments in mice, so again, we've moved from rats to mice, but the same process. So we raise these mice in either impoverished or enriched environments. What we see is that their cortical gene expression, so expression of genes inside the cerebral cortex, we can measure it at 2 days and 14 days after being in the enriched environment. Even 2 days after an enriched environment, you're seeing increased gene expression related to protein synthesis, protein processing, neural signaling, and neural growth. So this is making a major impact on what's happening within their brain just 2 days after being raised in this enriched environment. If you have any questions, send me an email.